So at this time, I would like to introduce our featured speaker, Dr. Anita Bhattacharyya. Dr. Bhattacharyya is an assistant professor in cell and regenerative biology. She received her PhD in anatomy and cell biology at the University of Cincinnati College of Medicine. She afterward completed a postdoctoral fellowship at Dana-Farber Cancer Institute at Harvard Medical School. And she also served as a scientist in the stem cell research program at UW-Madison's Waisman Center. So she has a substantial expertise in neural development, pluripotent stem cells. In fact, she's really the expert at Waisman working on stem cells and supporting that program. She also has expertise in human brain development, developmental disorders, neuron-glia interactions, and is working on Down syndrome, Fragile X syndrome, and autism. She recently received a very large award from the NIH that she's leading with a colleague. It's an $11 million transformative research grant from the National Institutes of Health that was awarded last fall to further understand cortical development and neuroplasticity in trisomy 21 using human-induced pluripotent stem cells. So that's quite the achievement and honor and phenomenally interesting work that she's doing. Her expertise in pluripotent stem cell generation, banking, and characterization is widely recognized. She serves as an advisor to companies, as well as serving on the International Society for Stem Cell Research, a task force on standards. Again, her expertise is just widely recognized. And we're also proud to call her a Wisconsin Alzheimer's Disease Research Center REC Scholar. And we're delighted to welcome her today to speak on modeling neurodevelopment and neurodegeneration in Down syndrome with stem cells. So please join me in welcoming Dr. Bhattacharyya.
So thank you, Barb. I want to thank you for that really kind introduction and also say how happy I am to be a REC Scholar. It really, as you heard from the introduction, my background and my training is primarily in development of the brain, and the REC Scholar Award really has enabled me to become more integrated with the ADRC and to learn more about both neurodegeneration more generally and Alzheimer's, and how my work, I think, can maybe fit in and maybe integrate some of the things that you all study. So I'm going to talk today about Down syndrome. And I think that this Down syndrome is familiar to most of you. Down syndrome is arguably the most complex genetic disorder. It is the most well-recognized form of intellectual disability. I think that most people in this room either know someone with Down syndrome or know of someone with Down syndrome because it is very common. It is, as I said, the most common genetic form of intellectual disability, with people with Down syndrome have IQs in the mild to moderate range. And the Down syndrome doesn't just affect the brain, it affects many other organ systems. It is a true developmental disorder. And the reason I'm talking about Down syndrome today is that with the increased life expectancy that individuals with Down syndrome have had in the last 20 or 30 years, has come the realization that individuals with Down syndrome are at a very high risk for developing Alzheimer's disease pathology. And I'll tell you a little bit more about that. I do want to point out two things about the cause of Down syndrome, which is trisomy 21. It's a triplication of the extra chromosome. And there are two things that are important about chromosome 21. It is the smallest human chromosome, but yet there are only over 350 genes encoded on this chromosome. And actually, I haven't checked yet, but with the most recent finishing of the Human Genome Project, there are probably even more now that the rest of the chromosomes have been annotated. So that's the first thing, that there are a lot of genes on chromosome 21. The second thing is that the genes on chromosome 21 are encoded on at least three different mouse chromosomes. And so this makes it very difficult, if not impossible, to model the genetic trisomy 21 in animal models. And so I'm not going to talk about animal models at all. The other aside that I'll bring up again is that there are specific genes on chromosome 21 that you may recognize, and I'll talk about later, may directly cause some of the characteristics of both Down syndrome, intellectual disability, and also Alzheimer's. So there are two features of Down syndrome that I'm going to talk about today. The first is one of the most obvious features of individuals with Down syndrome that relates to their intellectual disability, and that is the fact that people with Down syndrome have brains that are smaller, or we term this microcephaly, and they're about 20% smaller by volume than unaffected individuals. And what's interesting is whoops, I'm sorry, I turned the wrong one. Okay. There are certain areas of the brain, primarily the cortex and the cerebellum, that are smaller in Down syndrome. And this is due mostly to the fact that there are fewer cells, probably fewer neurons, but as I'll show you, we really don't know a lot about that. And the second characteristic that is relevant for this audience is the fact that people with Down syndrome develop Alzheimer's pathology. And so I'll talk about that in the second part of my talk. So my lab only uses human stem cells and human tissue to study Down syndrome. And so for those who may not be very familiar with human stem cells, I'm going to give you a quick 2-minute overview of human stem cells. Okay, so over the last 15 years, we've had this ability to generate what we call induced pluripotent stem cells, or I'm going to refer to them as iPS cells, from somatic cells from any individual. And so the way this works is that we are able to get skin or blood samples from individuals from any individual, whether they have a disorder, a disease, or unaffected individuals. And then we're able to reprogram them or transform them into cells called induced pluripotent stem cells. These induced pluripotent stem cells are iPS cells. We can then do genetic manipulation on if we want to using CRISPR-Cas9 editing. But what is important is these cells can turn into any cell type in the body, very much like human embryonic stem cells. And so in the lab, what we do in my lab is we're interested in turning these human stem cells into different cells of the brain. And so in the slide, you can see that we have the ability now to differentiate these iPS cells into different subtypes of cells in the brain, like different kinds of neurons. So for example, cortical excitatory and inhibitory neurons that we use to study intellectual disability, or dopaminergic neurons that are used to study Parkinson's disease, as well as other non-neuronal cell types. And we can then take those cells and do a lot of different kinds of analysis, really all of the things that everyone does in their wet labs. We can do transcriptomics, we can do proteomics, we can also assay them for their function and how well the neurons function, or other cells and how they function. And so when we put this all together, we have a really nice pipeline for understanding the cellular and molecular features of, for example, Down syndrome, because we can get blood or skin cells from individuals with Down syndrome, make trisomy 21 iPS cells, and then turn them into neurons to be able to ask specific questions, both about neurodevelopment, but also about neuron function. And so in addition to being able to generate different cell types, these iPS cells or stem cells are really good at modeling early brain development. So we know that the formation of the brain primarily happens prenatally, and this is an area that we don't have a lot of access to. But we know from both mouse studies and from the limited human studies that there is a very specific sequence of events that happen in order to build our brain, and that these sequence of events take place at very specific times during prenatal development. So for example, neurogenesis, or the generation of most of the neurons in our brain, happens between 10 and 20 weeks gestation. And then other events happen after that. And we know from many studies that have been carried out now in the last 20 years that our stem cells in the dish, when we differentiate them to different neurons, can recapitulate many of these steps in brain formation or neuron differentiation, and they do so with actually a very similar time dependency. And so we can use the cells both to study, again, specific neuron subtypes, but also neural development. And this is actually the focus of the grant that Barb was describing, is to try and use our trisomy 21 stem cells to see what is different during the formation of the brain in Down syndrome. So I'm going to start out by talking about what we have been doing to tackle this problem or this feature of Down syndrome, which is fewer neurons. So despite the fact that individuals with Down syndrome have smaller brains, there really isn't a lot of data to tell us, well, which cells are missing. And so this is a study, one of the few studies that was done, and it was done before many people in this room were born, probably, and so that tells you how old it is. And it was done in this study, they took post-mortem Down syndrome and unaffected cortex or brain and looked at how many neurons there were in the cortex. And as you can see from this picture, it's kind of hard to tell what cells are what using the methods that were used at the time. But nonetheless, they were able to show that looking at the graph, the black bars are control and the white bars are Down syndrome, that there are populations of neurons that are decreased in the brain of individuals with Down syndrome in adulthood. So we wanted to dig a little bit deeper to find out, well, what kind of cells are those? Because this really doesn't tell us very much. And so more recently, we've been using more modern methods of immunocytochemistry, which have been around for decades, to look at specific subtypes of neurons in the postmortem adult brain. And what I'm showing you here is three kinds of analyses that were done. The first is using an antibody to nu-N, which labels all neurons. And then below it, you can see the graph showing the difference between control and Down syndrome brain. And we really, very satisfyingly, were able to also show that there were fewer neurons in the cortex, in the brain. And in the second and third panels, columns, I'm showing you two different subtypes of interneurons that had been suggested to be decreased in the Down syndrome brain, but there was no data. And so what we've done is we used antibodies specific to parvalbumin, which is the most abundant interneuron in mouse brain, and then also looking at calretinin neurons, which are upper layer disinhibitory neurons. And what we found was no difference in the number or density of parvalbumin neurons in Down syndrome, but a decrease in the number of calretinin neurons. And so this helped us really define our question of, okay, we know this population of neurons are missing. Can we learn something about that, their development in our culture system? And so what we did was we took our trisomy 21 and our control iPS cells, we differentiated them to these specific calretinin neurons, and when we counted them in the culture, we found indeed we were able to generate fewer of these calretinin interneurons in the dish. And on the right is just the panel that I showed you before, showing that we can model this lack of neurons in our culture system, which I think is really critical because you may hear a lot about using stem cells to model different diseases, but unless the phenotype you see in the dish matches what is in the individual or into the patient, that really, then the studies may identify phenotypes that are more artifacts of cell culture. So we then took a step back and asked, well, why are there fewer of these neurons? And we specifically looked at a population of progenitor cells that we know give rise to these neurons, and also found a difference in the number of the progenitors. So if you start with fewer progenitors, then you will ultimately have fewer neurons. And so now we're looking at trying to decipher the mechanisms for why you have fewer progenitors, again, that leads to fewer neurons. And one of the things we've been doing, like everyone else, it seems, in the world, is to do single-cell RNA sequencing. And then this is also in collaboration with Daifang Wang, to cluster the populations of these progenitors in control and trisomy. And while they look fairly similar, we did find that there is a cluster that is in the trisomy 21 progenitors and not the others, suggesting that the development of these progenitors is different. And we may be, by looking at these clusters, we may be able to identify how it's different, more of the molecular and the cellular mechanisms. This is what we're doing now, looking at one type of neuron. Our goal is to look at lots of different kinds of neurons in the brain and study their development both in tissue and using our stem cells. So now I want to switch to talking about neurodegeneration in Down syndrome and show you this schematic, which I think to most of you in this room from afar looks very similar to what you've been seeing for many years, very similar to the JAX graph that you've seen. And in fact, it is very similar, showing that there is a progression this is showing Down syndrome only individuals, that there is a progression of amyloid accumulation and phospho-tau accumulation, and then onset of clinical symptoms, much like you would see in the general population if you were looking at Alzheimer's disease. But there are two important aspects of this graph for Down syndrome that I want to highlight. The first is if you look at the age on the x-axis, it's a lot younger than what you would see in the general population. And in fact, individuals with Down syndrome start to show amyloid in their buildup in their brains starting at around age 35 or 40. And this is based on much of the really nice work done here at our ADRC by Brad Christian and Segan Hartley and others at Pittsburgh. We're seeing a really early onset, and Down syndrome could be considered an early onset form of Alzheimer's disease. The other thing that's really important and really helps to inform our studies is the fact that it has been shown, again, from limited postmortem samples, that many of there are features of neurodegeneration more generally that can be detected in Down syndrome individuals' brains really early in their life. And actually, in fact, on the bottom, you may see that reactive oxygen has been detected in fetal development in Down syndrome. So this made me think that maybe using our culture system, we can find early events in AD pathology and also identify things that may make specific neurons more vulnerable than others. Because as you all know, that not every neuron may degenerate in the same way in AD or in Down syndrome. And so this is the area that we're now moving into. And so I did want to show, again, I want to show you that our cell culture system can actually recapitulate some of these events that are known to happen in people. And so the first is oxidative stress. So many years ago, we made cortical neurons from our iPS cells. We compared control and Down syndrome, and we did transcriptomics and found that there were large gene expression differences that in the Down syndrome neurons or trisomy 21 neurons, and the largest gene expression changes were actually in genes that were known to be involved in oxidative stress. So right away, this told us that, well, we're seeing the same thing. We're seeing this oxidative stress in fetal development. And we went on to show that the neurons that we generated have features of oxidative stress that we can measure at the cellular level. So on the top, I'm showing you a reactive oxygen that is in the cells. It fluoresces red when there's oxidative stress, and hopefully you can see that in our Down syndrome neurons, we have a lot more. And in the bottom is a measure of mitochondrial membrane potential, which is a measure of mitochondrial function, which is impacted by reactive oxygen. And I hope you can see that the two kinds of neurons do not look similar. And when there is increased, in this assay, if there is increased green fluorescence, then that means that the cell is, their mitochondria are unhappy. And so you can see that there's more yellow in the Down syndrome, more yellow-green in the Down syndrome neuron. And so again, we were able to validate these features that are known to happen in people with Down syndrome in our culture dish. And so now what I want to tell you about is kind of the direction we're now going in. And that is to study a different, to not study cortical development, but to actually look at a specific population that we've been particularly interested in, which are basal forebrain cholinergic neurons, which again, many of you are familiar with. These are a small group of neurons located in the basal forebrain in the nucleus basalis, and they constitute the largest cholinergic input into the cortex. So these are a small number of neurons that project to a lot of places in the cortex, and so you can imagine that if they are dysfunctional, they can have a lot of effects throughout the nervous system. And in fact, it's known that these basal forebrain cholinergic neurons are they do degenerate fairly early in Alzheimer's disease, and as I'll show you, in Down syndrome. And it's also, and so this is, and although this has been known for a long time, that these are a population of neurons that does degenerate, they really haven't been a focus of much of the research, I think, in AD. I think more has focused on cortical neurons. And in fact, there has been debate about how important these neurons might be in the progression of AD neuropathology. There have been the cholinergic neuron hypothesis, I think is about 30, 40 years old. Jose, tell me how many years? And where the loss of these neurons, as I said, was predicted to have, if this was an early event, would be predicted to have large influences on the health of neurons throughout the brain. This hypothesis and this, the involvement of these neurons, I think, has been going back and forth on how important they are. And so we decided to just jump into that and start looking at them ourselves. And primarily that is because in Down syndrome, there are fewer of these neurons in the adult brain. And so this provided us a way to ask the question, are there fewer neurons because they don't develop? Or are there fewer neurons because they degenerate very early? And so these are the kinds of questions that we're now starting to look at. And there is, again, one publication from 1985 showing that there are fewer of the basal forebrain cholinergic neurons in adult Down syndrome brain. And so we are trying to bring this information into the 21st century. And so I have a graduate student, Nicole West, and a postdoc, Matt Russo, who are working on looking very carefully and systematically at the cholinergic neurons in Down syndrome adult brain, with the hope that we will eventually be able to move that analysis earlier into younger ages, maybe prenatally, and also older in individuals who are aging and potentially showing features of Alzheimer's disease. And so we're excited about that, because no matter what the answer is, it'll be interesting, and that's always a good experiment, right? And so again, what we want to do, this is to try and find our baseline, what's really happening in individuals, but we also want to, as I said, we're going to ask whether there are a few cholinergic neurons, but also we can look in these tissue sections and ask if there are markers of degeneration or degeneration-associated markers or AD-associated markers. And then in parallel, our another graduate student, Jose Martinez, is trying to model the development and function of these neurons using our stem cells. And so what Jose is doing is taking our trisomy 21 and control iPS cells and differentiating them into basal forebrain cholinergic neurons, which is a little harder than making cortical neurons. And I'm hoping you can see we stain these neurons with beta tubulin, which labels all neurons, and choline-acetyltransferase, or CHAT. And you can see in green, and you can see that we get many of the neurons in our culture dish that are cholinergic neurons. And based on how we made them, we know that they are basal forebrain. And so with these cells in hand, we're asking very similar questions. Do fewer of these cholinergic neurons develop from our iPS cells? And do they have markers of degeneration and or dysfunction that we can measure in our culture system? And then ultimately, of course, what we'd like to do is compare what we can see in the tissue with what we can see in the cells. So I want to come back to chromosome 21 because I gave you that teaser at the beginning that there are some interesting genes on chromosome 21 that could very well and likely do lead to many of the characteristic features of degeneration or Alzheimer's disease pathologies that we see in Down syndrome. So there are many genes and they have functions in different cellular processes that can lead to dysfunction and death of neurons. So at the top of the list, I'm hoping you can see this, is APP. Everyone knows what APP is. Well, it turns out it's encoded on chromosome 21. And so individuals with Down syndrome have an extra copy of APP in all of their cells. And so you can imagine that if you have more APP, you have more substrate for amyloid production. And so that is thought to be the driving force of the AD pathology in Down syndrome. However, there is a large variability in how people with Down syndrome get Alzheimer's disease, when they get it, how severely they get it, how much it impacts their cognitive impairment. And so there are likely other modifiers across the genome that might also play a role. I will say, and I didn't mention this before, that on all of the transcriptomic works that have been done either in Down syndrome tissue or in cells, any kind of cell, there is not only increase of these chromosome 21 gene expression, but there are dysregulation of genes throughout the genome. And so it is not necessarily as simple as you have these extra copies of these chromosome 21 genes. There is immense regulation and interaction with genes throughout the genome. And so other genes that are encoded on chromosome 21 include DYRK1A, which is a kinase that phosphorylates almost every single protein in the cell, it seems. And so it can likely have a large effect on what's going on in Down syndrome. And then there are things like SOD1, which is involved in oxidative stress, and also others that we don't know very much about how they may be involved in neurodegenerative processes. And so in addition to taking kind of a more unbiased approach to see what is different about the Down syndrome neurons that we have in our culture dish and in tissue, we're also taking a more targeted approach by looking specifically at the impact of APP. And that is through, I just, again, want to remind everyone, although I know you know this probably better than I do, that the APP, or amyloid precursor protein, is a transmembrane protein, and it can be cleaved in different ways to either, if you go on your left, a non-amyloidogenic pathway and normal function, which we don't really know what the normal function of APP is right now, but there is something that it does that must be important that we will eventually learn that doesn't involve amyloid. And on the right, you know, as you all know, when it's cleaved in a certain way, you get a release of beta amyloid that can accumulate into plaques. So in collaboration with Subhajit Roy and Chris Saha, who is here, they came up with a strategy to stop the cleavage of APP into the amyloidogenic pathway as a way to test whether what effect that would have on different things in the cell. And I just want to show you from this paper that was published a couple years ago that in iPS cell neurons, if you use an antibody that recognizes the internal domain of APP. You can see on the bottom panel when we use this CRISPR-Cas9 gene editing to edit the APP molecule, then we can't see that extra part of the chromosome, so it's gone. And then what's more important is that by doing this, you can shift the cleavage of APP in the non-amyloidogenic direction. So you can see, So you can see in D here that in the control, you see some soluble APP, which would be the nice pathway. And when we do the gene editing, you see more of that pathway's products, the soluble APP. And concurrently, you see a decrease in the ratio of A beta 40 and 42. And so this is a really nice tool for us to try and get a handle on what is the role of APP in some of the cellular processes that we're studying, but without removing the normal function of APP that we know is there. So many people do knockouts to take out the whole gene, but this protein has other functions that we really don't want to perturb to get a cleaner experiment. And so this is work that I'm doing primarily with funding from the REC Scholar program. So just to summarize, I want to tell you that I hope I've convinced you that human stem cells are a valuable tool for studying both neurodegeneration and development, and using Down syndrome as a way to study some of these events because they can happen earlier in Down syndrome than they might in AD, and we can identify a population of people who we know will exhibit this pathology. And by using these stem cells, I've shown you that we are working on at least 2 different kinds of neurons. There are obviously many other kinds of neurons in the brain, and there are other neurons that are also susceptible in Alzheimer's disease. Another population that our collaborator Sushan Zhang is working on is locus coeruleus neurons, which are, again, a really interesting kind of neuron that not many people know much about. And so our goal, as I had said, was to identify both the cellular and molecular mechanisms that drive not only the altered development, but also that promote degeneration in these neurons. And so finally, I just want to show you, this is where my lab is at the Waisman Center on the far west end of campus. I want to thank people in my lab. I told you people who are working on some of these projects. And I also want to thank my funding and again reiterate that having this REC Scholar Award really enables me to move into looking at the degeneration in Down syndrome. And thank you. I'm happy to take questions.
So thank you, Dr. Bhattacharyya, for the presentation. So, so do a lot of other researchers, are they also currently looking into cholinergic neurons as well, or?
Not a lot, actually. They've been looking in mouse models, to some Down syndrome mouse models, but there isn't a lot of data in humans.
Okay, so like you're kind of looking into this right now, and.
Yeah, I know. As I showed you, we're starting kind of at the beginning. Okay. Like, okay, what do these neurons look like in brain tissue, postmortem brain tissue in individuals with Down syndrome?
Okay, thank you.
Okay.
Yeah, I was wondering if you can tell us too, again, the work on the cholinergic neurons. Do you think that's also going to help shed light on what is occurring in sporadic Alzheimer's disease?
Yeah, that's a really good question. I think it depends on how early of an event it is. And I've seen, again, this is a little outside of my field, but I've seen publications that say it could be a very early event. And then maybe that would suggest that that's an area to look at in more sporadic or in the general population. And I think what's been a difficulty in doing that is that most imaging technology, as I understand it, doesn't go that low. We tend to focus on the cortex, and sometimes you don't get a lot of the basal forebrain. We can, but many of the older images don't have it.
Right.
Yeah. And I think that would help us if we could look at
We
it.
can fix that.
I know. That's next year's grant, right?
Other question? Yeah.
Hi, Anita. That was a great talk.
Thanks.
And that gave me a lot to think about. You had mentioned that with the iPS cells, they kind of follow the same developmental progression into whatever they're going to turn into. Have you looked at, or do you plan on following these cells from stem cell to adult cell to see if there's like really early, like prenatal changes that can affect the adult cell to the point where it would cause a vulnerability to AD or something like that?
Yeah, that's a great question. Are we going to be following these neurons from really the development through? And yes, that's precisely what Jose is doing. So he is, as he's differentiating these cells from the iPS cells, he's finding, for example, that the Down syndrome cells are making less. And so there's one piece of data, but in addition, he's going to keep those cells going and look at them. So one caveat with this is that the neurons that we make from these stem cells tend to be young, kind of embryonic, fetal-like. They're not super mature, which is not surprising because they're growing all by themselves in a dish. And so they don't know how to grow up yet. And so that's one problem. There are ways that we are working to try and age them. This is an issue in the whole field, not just for us. So that's one thing we're trying to do. The other thing is that I didn't talk to you at all about we see these cells die. That's because we actually find it hard to see cell death in our culture because we've been spending 100 years trying to figure out how to keep them alive in the culture. And so to suddenly say, okay, now maybe they're too happy. We're supplementing them with too much. And so many other labs have tried to stress the cells to kind of induce that cell death. And so that's something we haven't looked at yet, but it's surprisingly hard to get them to die in culture.
Thank you.
Yeah.
All right, last question, please.
All right. I'll try to keep it brief then, Barb. Great talk. Thank you. So one of the things we see in the biomarker data, right, is that if you compare Down syndrome, autosomal dominant AD versus sporadic AD, right, you see really aggressive tau phenotypes relative to like the same amount of amyloid in both Down syndrome and in autosomal dominant AD. And one of the things that struck me from what you presented is that the density of the cells actually looks lower, right? Which from a biomarker and like PET perspective would mean it's harder to detect them. So like that just kind of got me thinking about this idea, right? And I don't know if you have anything that could like address this, but so you have this lower density of cells that could actually have tau tangles, right? But you have way higher tau in the biomarker data anyway. So I'm wondering if you have anything like planned that you would be able to kind of tease that apart, right? That like whether or not those phenotypes really are that much more aggressive, like the tau phenotype.
Yeah, I think that's a great question, and one that I haven't seen well addressed and not something we do. But you're right, is because I think that many biomarker studies take in or assume that people's brains are similar, right? And you're right, we have this population where there are gross differences in the brain. And so I think you may have to, for the Down syndrome population, have some sort of normalization or correlate the biomarker to something we know in the brain. It's a great question. And I think that, the field of Down syndrome AD or AD and Down syndrome is emerging and is very reliant on the AD field for sure. But I think they are finding that there are some differences that are going to have to be addressed.
Awesome.
Thank you.
All right, thank you, Dr. Bhattacharya, for a wonderful presentation.